afternoon, and also thank you for the opportunity to present our work here. So this afternoon I will be talking about 2-aminoimidazoles as a class of coatable biofilm inhibitors. And actually this research started in 2008 during my PhD, so this will become a 15-year story. So in my lab at the KU Leuven we study microbial community behavior, so mainly communities that contain pathogens, and we try to develop antimicrobial strategies that target microbial communities. And we are mainly interested in how the social interactions between microbes in these communities, their cooperation, their competition, how these interactions impact the virulence of the microbes, their resilience in the environment, and even their resistance evolution against antibiotics. And we try to apply this knowledge to develop novel types of antimicrobials. And much of this cooperation and competition between microbes actually happens within biofilms. So what are biofilms? Biofilms are surface-associated communities of microbes that are embedded in a self-produced exopolymeric matrix, actually a slimy layer. And these biofilms can attach to tissues in our body and cause infections, but they can also attach to different types of medical implants, such as heart valve prosthesis, as what we see here, but also on industrial surfaces and actually many surfaces that we have around us. One very important characteristic of biofilms is that biofilm formation is the production of EPS, exopolymeric substances. It are the EPS, these exopolymeric slimy substances that help the microbes to attach to surfaces, and these substances also provide a high tolerance to antimicrobials to the immune system. That's why many biofilm-related infections are very hard to treat. That's why many biofilm-related contaminations are very difficult to clean. So biofilm formation happens in a number of different stages. First, bacteria reversibly attach to surfaces. They can still move, and they can form clusters, after which they irreversibly attach, start to divide, and start to produce these exopolymeric substances. They can then grow out to mature biofilms that can contain complex structures such as mushroom shapes and water channels. And finally, bacteria can also disperse to colonize new surfaces. Now, because biofilms cause so many problems, we were actually interested in the development of anti-biofilm molecules. And to this end, we actually established a development platform for anti-biofilm molecules that has a number of different steps. So screening, chemical synthesis, mode of action, and application-oriented tests. And this is done with several collaborations, both within our university, but also internationally. And to illustrate this platform, I would like to focus the story of today on the 2-aminoimidazoles. And we identified these 2-aminoimidazole-based biofilm inhibitors so between 2008 and 2011 as a result of a high throughput screening of a big compound library. Can we get rid of the title there? A big compound library of more than 20,000 compounds, where we screened for anti-biofilm activity in a simple assay, and that's the Calgary biofilm device. So this assay actually just exists of a microtiter plate that's covered with a lid that bears PEGs, and the PEGs fit within the wells of the microtiter plate. We then add the nutrients, bacterial cells, and compounds to be tested to the microtiter plate. We add the lid, and biofilms are formed during 24 hours on the PEGs of the lid. We then stain these PEGs with crystal violet. Afterwards, we restain we destain in acetic acid solution, and then the optical density of the resolubilized stain is a measure for the amount of biofilm formed on the PEGs. So we screened 20,000 versatile compounds, and to put this into perspective, this means that we needed more than 3,000 biofilm plates, 800 rinsing plates, more than 1,600 crystal violet plates, and then destaining plates. So if you then know that every plate is about 1.5 centimeters high, this means that we ended up with a pile of more than 125 meters, which is just slightly higher than the highest cathedral in Belgium, in Antwerp. But it was worth the effort because we identified 140 hits that could inhibit biofilm formation. And after a number of additional screenings, amongst other at different temperatures, not only screening for prevention, but also destruction of biofilms, and testing not only against Salmonella, where we performed our initial screening, but also another pathogen, Pseudomonas, we ended up with 12 families of compounds. And here we purchased a number of analogues of these compounds to delineate early structure-activity relationships. And based on this, we ended up with 4 most promising families of compounds, and one of these families are the 2-amino-imidazoles. So here we see the imidazole ring, and here we see the 2-amino group. There are actually 5 aryl 2-amino-imidazoles because we always have an aryl ring at this position. So then we wanted to delineate the relationship between the structure, the exact structure of the compounds, and their activity. And to this end, we synthesized in collaboration with Professor Erik van der Rijken at our university 900 different analogues by now of these compounds, and they differ in the decoration pattern of the 2-amino-imidazole ring. So you see all the different positions where we have actually made alterations in the structure. And we then screened these compounds against a broad library of bacterial biofilms, monospecies, mixed species, containing different types of bacterial and fungal pathogens. And we also screened the compounds in a number of simple cytotoxicity tests. And what we found, and this is actually interesting, is that the activity spectrum turned out to be tunable. So we can actually optimize a certain compound to target a certain species in a certain application. So to give an example, we found that the N1 substituted compounds have a very broad activity spectrum, but also were toxic in our cytotoxicity tests. 2N substituted compounds target more narrow spectrum of gram-negative bacteria and fungal species, but they were not toxic in these tests. And if we then had substituents at the 2 positions, we found activity against gram-positives, but not gram-negatives, fungal species, and they were also non-toxic. Then we actually asked, how do these compounds inhibit biofilm formation? What is their mode of action? And to unravel this, we first made an overview of all the regulatory processes that were known at that time to be involved in biofilm formation by Salmonella. Because biofilm formation is highly species specific, we focused on Salmonella. And we then actually selected genes at strategic positions in the network, the ones with the green circles. And we took the promoters of these genes and pasted them in front of a promoterless GFP gene, and that encodes a green fluorescent protein. So by measuring green fluorescence, we could find out which biofilm-related genes were actually affected by our compounds. And this way, we found that they imidazoles activate the expression of FOPQ and its regulon. FOPQ, it's a membrane-based receptor in Salmonella, that this resulted in the delayed activity of RPOS, which is the general stress response system. And this finally, and that's most important, resulted in an inhibition of the transcription of CSGD. Now, what is CSGD? CSGD is the master regulator of these EPS components, these slime components of the matrix in Salmonella. So it regulates both cellulose and curly fimbriae, which are the most important EPS components in Salmonella. And we then showed that this inhibition of the EPS, so it targets EPS, does not only reduce the biomass with crystal violet, but also the number of cells on the surface and the tolerance to antibiotics and disinfectants. So this brings us to the following working model. Biofilm EPS is important for bacterial attachment and tolerance to antimicrobials. If we apply our 2-amino-imidazoles, they act as EPS inhibitors, reduce attachment, reduce tolerance to antimicrobials. So this means that it would be interesting to use these compounds in a preventive way, maybe as coatings. But as for all types of long-term preventive antimicrobial strategies, resistance could be a major concern. And that's why in the next step, we actually wanted to focus on evaluation of resistance development against these 2-amino-imidazoles. And at that time, I was actually doing a postdoc in the lab of Kevin Foster in the University of Oxford, and he's an evolutionary biologist interested in the evolution of cooperation and competition. And actually, at that time, I realized that these EPS might actually be a form of cooperation between microbes, that they actually might be a type of public good. And that's important, as I will tell you later. What are public goods? That are substances that are costly for individual microbes to produce, but that are shared between the individuals and provide a benefit at the group level. You can actually compare this with our tax system, costly for us as individuals to pay, but we share the money, build roads, build schools. So public goods in bacteria are similar. Well, why is this actually interesting that these EPS compounds can be public goods? Well, this is because social evolution theory predicts that inhibitors of public goods would not select for resistance, but against resistance. Yeah? So if you apply a public good inhibitor, there's no public good, population of microbes is weakened. Suppose now that a resistant microbe emerges, then we actually can expect that the sensitive microbes, the green ones around, also have access to this public good, yeah? But actually do not pay the cost for the production, the energy cost for the production of the public good. So these sensitive microbes actually behave as cheaters. They exploit the costly public good produced by the resistant bacteria. And that's why the sensitive bacteria might have a net advantage, grow faster than the resistant bacteria, and outcompete the resistant bacteria. So if we would apply this theory to our biofilm or EPS inhibitors, this would mean that, again, if the EPS inhibitors inhibit biofilm formation, if you would get resistant bacterium that emerges, that this would actually share the slime, the EPS, again, with the sensitive microbes. Sensitive microbes might act as cheaters, exploit EPS without paying a cost for it, and grow faster than the resistant microbe. So there would be a selection against resistance, not for resistance, as we see with classic antibiotics. This is, of course, all nice in theory, but we wanted to test this in the lab. So our first question was, is Salmonella biofilm EPS exploitable? Can bacteria do not produce EPS, so that would be sensitive to an EPS inhibitor? Can these bacteria exploit, make use of the matrix produced by a resistant bacterium? So to test this, we performed competition experiments between a wild-type EPS producer strain and our delta CHGD EPS non-producer strain. We mixed them in different ratios and let them form biofilms on the bottom of Petri dishes. We then poured off the planktonic cells above the biofilm and scraped off the biofilm cells. And then we counted both the EPS producer and non-producer who were labeled with different fluorescent labels. What we found was that in monoculture, the EPS producer has a higher fitness than the EPS non-producer, which we already knew. But when we look at all the combinations of strains, we see that this inverses and that the non-producer actually gets a higher fitness than the producer, which is in line with the idea that this non-producer is actually benefiting and exploiting the EPS produced by the producer. So this would actually indicate that we would have a selection against resistance. But then we also looked at longer-term competition. So we did not do one round of biofilm formation, but we also always inoculated the scraped off biofilm again, and this for 17 rounds. And what we found then was that there was a decrease in the frequency of producers in function of time till a very low amount of EPS producers. So the EPS producer is actually almost completely outcompeted after 34 days. So this was very interesting. We also wanted to further validate this with microscopy. Here you see that the EPS non-producer just forms a sparse layer of cells on the surface. But if we then add 10% of EPS producer, this EPS producer forms the matrix and has a three-dimensional structure. What you can see in this image is that the non-producer, in fact, localizes at the same locations as the EPS producer, which further indicates that the non-producer is exploiting the EPS produced by the producer. So this is a first indication that probably we can have a selection against resistance. So we now want to test whether we actually saw resistance emerging or not. So to this end, we performed an evolution experiment. So we inoculated only a sensitive strain, but now in the presence of the EPS inhibitor, let them again form biofilms. We only scraped off the attached cells and then re-inoculated. So in this way, we had a selection pressure for biofilm formation. And we did this for 40 days. And what we observed was that when we compare the endpoint of the evolution experiment with the starting point, that the endpoint strains were still as sensitive to the EPS inhibitor as the original parental strains, the non-evolved strains, but we have the same reduction in biofilm cells and that CHGD expression is still inhibited. So we did not see signs for resistance evolution after 40 days. If we apply classic antibiotics in the same setup, we see resistance after about 4 days. So we didn't see resistance, but then we wanted to validate whether this absence of resistance is really because you have a counter selection of the resistant microbes. So to test this, we wanted to compete a resistant and a sensitive strain in the presence of an EPS inhibitor. To that end, of course, we needed to identify first the resistant strain. So we performed a screening of a knockout library of 4,000 strains, did not find the resistant strain. Then we went to natural isolates, and there we found a combination of strains where one strain was more resistant to the EPS inhibitor and another one less resistant. We again competed those strains, and we found that, per definition, of course, the resistant strain had a higher fitness than the sensitive strain in monoculture. But if you combined both strains, this inversed, and the sensitive strain outcompeted the resistant strain. We also looked at longer-term competition and found that actually there is a selective enrichment of the sensitive strain in function of time, so resistance is counter-selected, exactly the opposite as what you see in case of a classic antibiotic. So this was, of course, very encouraging, and that's why we actually were motivated to look into a number of applications. And the first application was application of a covalent anti-biofilm coating with these inhibitors on orthopedic and trauma implants. And this was partly done in the frame of an FP7 EU project coating. So again, in collaboration with Erik van der Rijke and also a company called Hemotech, we made a covalent coating of the imidazoles on titanium implants, little discs in fact, where these compounds were linked via an amide bond and an organic linker to the titanium surface. Then first tested the activity first in vitro against Staphylococcus aureus biofilms, which are important for these types of implants. And with confocal microscopy, we could see that the anti-biofilm coating decreased the area covered with microbes with about 70% compared to control discs. We then wanted to use a simplified in vivo model for infection of implants. It's a subcutaneous model in mice. So the disc is at day 1 implanted subcutaneously in the back of the mice. At day 2, there's an injection of about 10 to the 8 cells of Staphylococcus aureus. And at day 6, animal is sacrificed and the implant is explanted, and then we can count the microbes on the implant. And we recovered 25 times less Staphylococcus aureus cells from a coated implant compared to a control implant. Of course, when we have such a coating, it should not only inhibit the number of bacteria, it should also still support osseointegration and do not inhibit the osteogenic cells. And that's why, in collaboration with the company Edicel, we tested the impact of these compounds on the proliferation and survival of human bone marrow-derived stromal cells. We found that the coating supported attachment of these cells and also the growth. We even saw a slightly higher growth of the cells compared to a non-coated disc. To further validate this, we also looked into a rabbit model for osseointegration. So this was done in collaboration with the University of Ljubljana. So now we coated small cylindrical discs with the compound. Discs were implanted in the femur of rat. A healing period of 8 weeks was allowed, and then X-ray tomography was used to study osseointegration. We found that the volume and the distribution of the bone phase around the implant and the coated implant were comparable. So that's a further validation that the compound does not affect osseointegration. We also moved into a completely different application because these compounds have very broad activity spectra, so that's interesting in terms of types of applications. And we looked at biofilm formation in irrigation water system in collaboration with a number of horticulture research associations in Flanders. Especially problematic in these greenhouses are infections with Agrobacterium rhizogenus. So these bacteria can form biofilms on the kilometers of pipelines in greenhouses and can then infect the roots of the plants. And this causes an excessive uncontrolled root growth, but reduces the production of the plants, so reduced tomato yields. And we used actually in vitro experiments, pilot scale water systems, and a real large-scale greenhouse trial to show that our inhibitors inhibit biofilm formation of this bacterium and actually also the symptoms within the plants. So we have a lower occurrence of hairy root disease in these greenhouse experiments. In the frame of a meeting of the AAA Code project, so a European project where we're actually attaching 2-aminoimidazoles to nanocellulose coatings in order to treat surfaces on high trafficking materials such as buses, airplanes, and so on. So to summarize, 2-aminoimidazoles have a broad spectrum biofilm preventive activity. They are tunable in terms of their spectrum. They inhibit biofilm EPS production. They are evolutionarily robust because EPS is a public good. They support osseointegration and bone healing, and we can translate our in vitro activities to in vivo and in situ models. So a lot is still going on, and I'm very excited for the future. I would like to thank a number of people, Professor Van der Rijken, who performs all the chemistry of the 2-aminoimidazoles and the coatings, Professor Foster, with who I did the study on evolutionary robustness. My own lab, the MICA Lab, with a special thank you to Guglielmo, who is also here, and of course the AAA Code Consortium, and especially Samuele for organizing this nice meeting. Thank you.